Hi guys, welcome to Mastermed Academy. Today we'll be talking about adaptive immunity. I have made two parts of adaptive immunity. This is part 1, and in part 1 we'll be discussing about the major components of adaptive immunity, what is an MHC molecule, what are its function, and we also will be talking about VDJ recombination regarding the T cell receptors and antibodies. What exactly is the process? I will be breaking it down. Also, we'll be talking about B cells and what are the functions of B cells and what is class switching and what is its significance. So let's start. So today we'll be talking about adaptive immunity part 1. Now this is a beautiful picture of a T lymphocyte. Now this picture is taken using a scanning electron microscope, and this is a very beautiful picture if you ask me. This is how a T lymphocyte looks. It's like a 3D picture. Now what is an adaptive immunity? Now adaptive immunity consists of T cells, B cells and antibodies that are already circulating in our blood. Now the response is initiated by the recognition of antigen by antigen receptors of lymphocytes. So here is the antigen receptor interaction and the production of antibody business here. Now adaptive immunity is highly specific and it develops over a long period of time. Unlike innate immunity, as I mentioned earlier in my innate immunity lecture, that innate immunity is non-specific and it is very rapid in response. Now memory response is fast and is refined over time. Now memory response is a part of secondary immune response. We have primary immune response. We also have secondary immune response. Now primary immune response is basically when our body encounters the antigen for the first time, and it takes a long time to produce the specific antibody towards that specific antigen. So that is the primary immune response, or in other words, we can say the first encounter. Now when we have the second encounter with the same antigen, the memory cells which are already present in our body will be helping the adaptive immune system to recognize the antigen quickly, and that will lead to the quick killing of the pathogen. That is why I say that secondary immune system largely depends on the memory cell. Now microbial resistance is not inheritable, unlike innate immunity. VDJ recombination is seen during lymphocyte development. Now what is this VDJ recombination business and what does it stand for? Why do we require a VDJ recombination? I'm going to be talking about that in the next slide. Now we have a B cell, of course, and B cells are responsible for antibody production. Exactly speaking, plasma cells are responsible for antibody production. Now this is a picture that explains the VDJ recombination. Now VDJ, V stands for variable region, D stands for diversity region, and J stands for joining region. Now let's talk why do we require a VDJ recombination and what is its significance. Now VDJ recombination is basically a game of genes where we shuffle some genes, play with the genes, and that leads to production of different type of receptors. Now they could be T cell receptor, they could be different type of antibodies. Now why do we need this diversity? Because we have so many types of antigens which are present in this whole world. Now of course we don't have a receptor for each of those antigens that are present, right? So with this process we form diverse receptors for the antigens, for the different antigens that are present in this whole world, Now if you know the structure of an antibody, then we know that we have a heavy chain and we have a light chain. Now this region, let me name this as L and H. L stands for light chain and H stands for heavy chain. Now this region is known as the FAB region. F stands for fragment and AB region, or AB stands for antigen binding. Now this structure here is named FC portion. Now C stands for constant here. Now when we are talking about VDJ recombination genes and the process, we are basically talking about the FAB region. Now here we are trying to make diverse FAB portion of the antibody. So we have different types of FAB region here, which will allow us to bind different antigens which are present in the outside world, correct? So that is the reason why we require the VDJ region. Now another point to remember is that in this VDJ recombination, there is no change or there is no effect in the FC portion of the antibody. For this FC portion of the antibody, we have a different process. But right now, VDJ recombination is for only FAB region, because we want different type of FAB regions so that we could bind different antigens. Now, FC portion is actually responsible to determine what kind of antibody do we have. So, is it IgM, is it IgG, or IgA, or it could be anything else. Now, as you can see in the cartoon here, we have 4 V genes or 4 boxes here, and then we have 4 D genes, and then the same with the J gene, and then we have a constant gene. Now in VDJ recombination, only the VDJ genes are shuffled. The C portion is always constant, and as you can see in this cartoon, it is not even touched. This C region is what will determine if the antibody is IgM, IgA, IgG that I mentioned previously. Now, when the recombination starts, the first step is to join the D and J gene. So as you can see, we remove the unwanted D and J gene segment, as you can see here, and then we join the D and J gene here. Then after that, the second step is the removal of unwanted V genes. So we remove the unwanted V genes, and then this will lead to recombination of V and the DJ gene, marking them as one complex, VDJ. Now the last step is to join the constant that we did not touch to form the complex VDJC. Now this is how the FAB region is formed, and the FC portion is left constant. And this shuffling takes place in many ways, so we have various types of combinations, but I hope now you understand why do we require this VDJ gene. Now how does it help us to form different FAB regions, and I hope you understand this now. And before I move to the next slide, I would like to tell you guys that when I told you that we had the process of recombination, then we had the process of deletion, then again we had the last step, which was the recombination. Now this deleting, combining process is mediated by a special gene, and the name of that gene is called RAG gene, specifically RAG1 gene. Now this is high yield to know for your step exams, because RAG1 gene is responsible for the recombination and deletion and everything. So if we don't have a RAG gene, we have a problem with producing diverse receptors for the antigens out there. So we also have a disease called RAG1 disease, in which we have a mutation in the RAG gene that leads to basically not producing various types of receptors for the antigens. And the RAG1 disease is basically an autosomal recessive disease. So I hope you guys understand now the VDJ recombination. Now the next slide basically shows the process of adaptive immune system and how it responds to a specific pathogen. Now we have a macrophage here and the pathogen nearby, and the pathogen in our example is a virus. Now the pathogen will be engulfed by the pathogen will be engulfed by macrophage through a process called phagocytosis. Now after phagocytosis and breaking the pathogen in the phagolysosome, the macrophage will show the receptor on its cell surface for a nearby T lymphocyte to be recognized. Now a T lymphocyte will recognize the antigen which is presented by the macrophage, and it will lead to activation of either natural killer cell or cytotoxic T cells. As I mentioned that the pathogen is a virus, so whenever we have a viral antigen expressed, we always have either a natural killer cell acting towards it or a cytotoxic CD8 positive T cells. Now cytotoxic T cells will destroy the infected body cell and get rid of the virus. On the second step, we have a B lymphocyte formation, and that will lead to proliferation and formation of plasma cells that will produce specific antibodies towards this viral antigen. Now, the antibody will coat itself around the viral pathogen, and then a nearby macrophage will see this virus coated with the antibody, and then it will lead to phagocytosis and digestion of the viral complex. So I hope you understand this cartoon as to why and how the adaptive immune system works. You can also see down here in the image that the memory cells are also formed in the primary immune response, and memory cells will later induce the secondary immune response if it encounters the same pathogen. So I hope you understand this diagram. It's really simple. It's not so complicated. So let's go to the next slide. Now, major histocompatibility complex, short form is MHC. They are proteins present on antigen presenting cells that will display the peptide antigen. Now you have to remember that if you want to display any antigen on MHC molecule, it has to be a peptide antigen. It cannot be a raw antigen, let's say. It always has to be a protein. If you don't have a protein antigen, the T cell will not recognize the antigen, right? That is why I say in my first line that membrane proteins on antigen presenting cells display peptide antigen for recognition. MHC is encoded by HLA gene. Now HLA stands for human leukocyte antigen, and it is located on chromosome number 6. Very important to know. MHC locus contains two sets of highly polymorphic genes, which is named as class 1 and class 2 MHC genes. Now I'll be talking in detail in the next upcoming slides about MHC class 1 and 2. Now this MHC has a major role in transplantology. Now, properties of MHC genes. Now what are those? These genes are very polymorphic, highly polymorphic. Second, these MHC genes are codominantly expressed, meaning that the alleles inherited from both parents, mom and dad, are equally expressed. Now class 1 molecules are expressed on all nucleated cell. Very important to know. That means it will not be expressed on RBC. So the exception is RBC, because RBC don't have a nucleus. Class 2 molecules are mainly present on APC cells. Now they could be dendritic cells, macrophages, and B lymphocytes. Now this is a beautiful structure of MHC class 1 and class 2. As you can see, we have 2 alpha chains and 2 beta chains in MHC class 2 molecule, whereas we have 3 alpha chains in MHC class 1 and a beta microglobin in MHC class 1, right? So I hope you can understand the image and refer to it. Now, what is the difference between MHC class 1 and class 2? This is very important to know as well. Now, MHC class 1, how I remember is, it has only one letter. So the 1 stands for one alphabet. So what kind of HLAs do we have? So it will be actually HLA, it will be HLA-B, it could be HLA-C. So only one alphabet. So MHC 1 stands for one alphabet. MHC 2 stands for 2 alphabets. Simple, right? So we have HLA-DP, we have HLA-DQ, so we also have HLA-DR. So now I hope you guys can easily distinguish between the 1 alphabet and the 2 alphabet, meaning you can easily differentiate if this molecule belongs to MHC class 1 or MHC class 2. Now, the next point is that the MHC1 is having TCR and CD8 binding process. MHC class 1 always binds CD8 positive cytotoxic T lymphocytes, whereas MHC class 2 will always bind a CD4 helper T cell. So let me write T helper here and T cytotoxic here. In MHC1, the antigens are endogenously synthesized. I will explain this in the next slide exactly what I mean by this. Whereas the antigens are synthesized exogenously in MHC class 2. MHC class 1, we have at the antigen loading in rough endoplasmic reticulum via a transporter protein called transporter associated protein, TAP. So TAP stands for transporter associated protein. Whereas in MHC class 2, we have an antigen loading that is done in an acidified endosome. Now we have MHC1 molecule in which we have beta 2 microglobin, whereas in MHC2 we have an invariant chain. So this is the beta microglobin chain in MHC class 1 that I talked about. Now this is a picture that shows us how the antigen is endogenously processed and shown in a cell that has an MHC1 receptor. So let's have a look at this. So let's imagine that this is a protein here and it is gonna go through a molecule that is going to break it into pieces. Now that molecule is called a proteasome. So proteasome is a molecule or is a complex that actually breaks proteins using enzymes such as proteases, and it will break the peptide bonds between proteins, and this will result into small protein pieces known as peptides. Now these peptides will actually be transferred or thrown through the TAP molecule, which meant the transporter associated protein, and it will reach into the endoplasmic reticulum. It will bind the MHC class 1 molecule. It will be processed in the Golgi apparatus here, and then it will be packaged into a secretory vesicle which will go up in the cell, and then it will be expressed as a peptide on MHC class 1 molecule in the surface of the cell. So this is how the antigen is endogenously processed and presented on the cell surface. So I hope you guys understand this. Let's talk about B cells. Now B cells belong to humoral immunity, very important to know, whereas T cells, they belong to cell mediated immunity. Both of them are a component of adaptive immunity. Now B cells develop from hematopoietic stem cells derived from bone marrow. Additionally, B cells act as antigen presenting cells, and they will present the antigen to a T cell, and that would lead to secretion of cytokines. Now B cells undergo class switching and secrete appropriate antibodies against pathogens. Now what is this business of class switching? I will be talking later in the presentation. Now this is a beautiful picture of a B cell. This is a 3D picture, and I think we can take a second to appreciate the picture, how beautiful it is, and it's really cool. Now this slide is very important for your step exams. Now this slide tells us about the B cell surface proteins, which you have to know for your tests. Now cell surface protein CD40 will bind CD40 ligand on T cells, and this binding leads to the antibody class switching. Now class switching, in one sentence if I have to say, it means going from IgM to, let's say, IgG, or going from IgM to IgA, and so on. It could be IgE. This is class switching. Now MHC class 2 molecule will bind the T cell receptor where the antigen is being presented. Cell surface protein B7 will bind the CD28 on T cell, and it will produce the cytokine. Now the cytokines can be used in the process of inflammation and other processes. Now pan B cell markers. Now pan B cell markers are basically CD19, which is present on all B cells, CD20, which is present mostly on B cells but they are not present on plasma cells, CD21, and they are useful for complement, and EBV. EBV stands for Epstein-Barr virus. So we have CD21. Now class switching. Now what is class switching? Now class switching is a mechanism that changes a B cell's production of immunoglobulin from one type to another, such as IgM to the isotype IgG. Now initially when activated B cells are made, they produce in small amounts IgM. So you can remember that whenever a B cell is activated, it initially produce small amounts of IgM and IgD, right? Now during this process, the constant region portion of antibody, that is the heavy chain, is changed, but the variable region of heavy chain stays the same. Now let's make a diagram of the antibody and explain it better. L stands for light chain and H stands for heavy chain. I mentioned previously that this is the FAB region here, right? And this is the FC portion here. And I told you when I discussed about VDJ recombination that it is responsible for bringing diversity in the FAB portion of the antibody, which actually affects the antigen specificity. But class switching will act, or basically is responsible for FC portion of antibody. Now, the FC portion is the only portion that is changed in this process, not the FAB portion. Now, since variable region does not change, class switching does not affect antigen specificity, as I mentioned. Post class switching, we have IgG, IgA or IgE antibodies, not IgM, because I told you, first we make either IgM, which is like in more amounts than IgD. So it's IgM and IgD initially, then after class switching we have IgG, IgE or IgA. So I hope you understand this. Now this is basically the process of class switching here. Now this cartoon actually shows how it's done. Now this is the VDJ gene that was specific for the FAB portion of the antibody. Now this will remain unchanged. This will remain constant throughout the process. Now we will be breaking down the constant portion or the constant gene, as you can see here, which is named mu, delta, gamma 1, gamma 3, gamma 2a, epsilon and alpha. Now this mu stands for M. So if you have the joining of the mu portion with the VDJ gene, then the antibody will be called IgM. If you join the gamma 3 portion with the VDJ gene, then the antibody would be called IgG3. Same with epsilon, it will be IgE, and alpha IgA. So I hope you understand this. It's very simple, right? Now you remove the DNA segment by enzyme activity, and then through a process of non-homologous end joining of the DNA, you basically put the constant gene and combine it with the VDJ gene. Now in this example, we combined the gamma 1 gene with the VDJ gene. So now this antibody would be called IgG1. So I hope you understand the class switching mechanism, why it's important, what is it for, and how the process works. Now, what are the triggers for class switching? We have cytokines, interleukin 4 and interleukin 5, very important to know. And we have a CD40-40 ligand binding. These are the triggers for class switching. Now, B cell activation. Now, there could be two types of activation of the B cell. It could be T cell dependent or it could be T cell independent. Now, T cell dependent binds peptide antigens. I already mentioned that if you want the T cell to recognize the antigen, it has to be a peptide antigen. Whereas T cell independent will actually bind the non-peptide antigen. The non-peptide antigen could be an LPS, like a lipopolysaccharide. That's non-peptide. It's not peptide antigen. Belonging to which bacteria? Gram-negative bacteria. T cell dependent has a strong response. Independent has a weak response. It promotes the B cell proliferation, class switching and somatic hypermutation. Whereas the T cell independent is mostly IgM, little bit of IgD, but no class switching is involved here. Rather, we have toll-like receptors. Toll-like receptors were belonging to which part of the immunity? Innate immunity. T cell dependent has a strong memory response and T cell independent has a weak memory response. Now, this is a structure of a T cell dependent B cell activation, and we can clearly see that there is a CD4 T cell here that actually is bound with the antigen which is presented by the MHC Class 2 molecule, and that leads to a cascade of processing and production of antibody. So this is T cell dependent B cell activation. Now, what is the fate of B cell? Post activation, the B cells will become memory cells, which is responsible for secondary immune response. So this is for the future fighting. And the second is the plasma cell. Now, plasma cells makes antibodies. I mentioned it previously. So either the B cell is a memory cell, which will help us in the immune response, secondary, or it makes plasma cells, which will make the antibody and help us fight against the pathogen. So I hope you understand the B cell. This is the process, the whole process, and I try to make it simplified for you guys. I hope you enjoyed this video. Please don't forget to like, share and subscribe to my channel. And if you have any questions, please write us in the comments and I will try to reply as soon as possible. And thank you for your attention. Bye-bye.